Good afternoon, everyone. A very warm welcome from all of us here in the Faculty of Science and Technology at the University of the West Indies, Mona campus in Jamaica, to all of you who are viewing in Jamaica, the wider Caribbean, and the world. A very special welcome to the students in the audience, those of you who are from primary schools, secondary schools, and/or university students who have an interest in science and who want to use it as a tool for solving problems like COVID-19, or who are just here to get inspired so that they can get better grades in science. I want to welcome the policymakers and the business leaders and encourage all of you to make your contributions to this discussion, as science must hold hands with policy and business before it can create wealth and impact lives. To other members of the public who are using this as an information session, we say welcome. Please feel free to share your thoughts, as you are important stakeholders in the UWI, and FST, the Faculty of Science and Technology, values your partnership. The COVID-19 pandemic is the worst global crisis since World War II, and it currently threatens to eliminate the social and economic advancement that many countries, including Jamaica, have enjoyed in recent times. However, like it has done throughout history, scientific knowledge is guiding humanity through these unprecedented challenges, from characterizing the virus and sequencing its entire genome, to understanding how it is transmitted and how we best protect ourselves from it. Science is fulfilling its classical goal of building knowledge and understanding Good afternoon, everyone. The Faculty of Science and Technology at the University of the West Indies, Mona campus, the FST, welcomes you to Forum 2 in our special Science for Today series, Fighting COVID-19, Science in Action. Now, after Forum 1, we received numerous positive comments from you. You indicated that you found the information presented on screening, testing, tracking, and protecting yourselves from COVID-19 and the misconceptions concerning COVID-19 extremely useful. Many of you sent commendations to our researchers and our students who have coordinated their own response to COVID-19. Your feedback, overwhelming, endorsing the Times Higher Education rankings, which put the UWI in the top 1% of universities in Latin America and the Caribbean, in the top 1% of Golden Age universities, and which names UWI as the #1 university in the Caribbean. The FST has played a vital role in the UWI achieving this triple first ranking, and we proudly claim this position in the Caribbean as the go-to place for science knowledge, science research, and science solutions. Today, we further this mission with an impressive lineup of speakers. Our mission fulfilling both the classical role of science to build knowledge and understanding and its modern mission to make people's lives better. So, stick around, ladies and gentlemen, for the next 2.5 hours, from data science and ethnomedicine to misconceptions about the wearing of masks. Are you safe because you put on a mask? We are talking COVID in our water, in our wastewater, and our drinking water. What should you know? We show how partnerships among teams of scientists can lead to real solutions for developing countries like Jamaica, and we showcase innovation from the FST students who are in action against COVID-19. This afternoon, there is something for everyone. And with the number of COVID cases in Jamaica rising sharply, we will all emerge from this forum empowered to put science in action for ourselves and fight COVID-19. So I urge you to call someone and tell them to log on. We invite you to send your questions in at any time during any of the presentations. My colleague, Dr. Kimberly Stevenson, is moderating the live chat, and speakers will be available to respond to your questions and your comments. Now, before we get into the substantive matter, allow me to make 2 minor adjustments to the program. First, Dr. Tanya Kerr is unable to be here today, and so the talk on material science combating COVID-19 will be canceled. But not to worry, ladies and gentlemen. I think we have enough matter to keep you excited, to keep you interested, and to keep you engaged. Second, Dr. Gunjan Mansingh has also been unfortunately delayed, so her talk will be replaced with a presentation on a related talk by Dr. Ricardo Anderson entitled Data Science Aiding COVID-19 Management. Now, to the first talk, Dr. Ricardo Anderson. Dr. Anderson is a lecturer in the Department of Computing here at the UWI in the Faculty of Science and Technology. He lectures in data science and software engineering. He holds a PhD in computer sciences from the UWI, a bachelor's and master's degree in computer science and information systems, respectively, and he's also qualified as a project management professional. Dr. Anderson researches knowledge management systems, knowledge discovery in databases, and data mining and decision support systems. Prior to becoming an academic, Dr. Anderson worked in IT management and software development for a few years. Ladies and gentlemen, please help me to welcome Dr. Ricardo Anderson.
Hello, my name is Ricardo Anderson, and my talk today is about how data science is aiding in the management of the COVID-19 pandemic. Okay, so before I get into the details of how data science is actually being applied in this pandemic or to assist in the management of the pandemic, I want to talk about the multidisciplinary nature of what data science really is. So it really requires a number of things. Computer science, we need data processing and retrieval, we need the algorithms. There is some statistical research and some broader application of mathematics. Machine learning, and of course, domain expertise is required. And so teams that are engaged in applying data science in any particular domain, and in this case, it is in the domain of disease management, will have to apply several technical skills to the particular domain in order to get the necessary results. And so, what is the real aim of data science in the main? The real aim is about discovering patterns, getting inferences, and being able to predict or understand some phenomenon. And so in this case, the domain expertise really is with the epidemiologist and other medical professionals, and they are the ones who will help to guide us in applying these techniques in order to get value from the data which is collected about the pandemic. So before we can process data, of course, we need to collect that data. And so we talk a little bit about where the data comes from that will allow us to use data science, or that has been used in data science. So before we can actually process data, we need to acquire that data. And so I'll talk a little bit about the data sources for pandemic-based data in relation to COVID-19. So what we have seen is an emergence of data collection applications because, of course, this era is dominated by the use of mobile devices, and everybody talks about an app today. And an app is just a small piece of software that carries out some function that is relevant to the user or some population. Now, many applications have been developed to collect, process, and manage data related to the pandemic. We find also that many of the institutions that are collecting data about test results, signs, symptoms, or other disease details that will allow them to be able to make inferences, they use these software to collate and process their data. Incidentally, of course, we know that a lot of mobile devices run on the Android operating system, which has been built by Google. And Google has their Play Store. And usually, in order for an application to become an official part of the Play Store, it would have to go through a review process. And in light of the COVID-19 pandemic, Google has released a policy that now will expedite that process of review so that applications that meet certain criteria that they have set can actually make it more quickly into the Play Store so it becomes available for people to use. So this is a global fight. And of course, it is driven by data. Now, there are many other applications that have been developed. So for example, in Spain and in other parts of the world, we have seen applications that provide for reporting and managing symptoms. So we have questionnaire-based input of symptoms, and the user, for example, of Assistencia COVID-19 will be able to get guidance from that application based on what they have input in a series of questions within that app. Then the app also provides recommendations regarding the need to either isolate or contact health services based on the user's answers to the questions. These apps also allow for tracking of personal symptoms as they evolve. So, for example, the user can put in symptoms on a particular day, update that, on another day, and over time that tracking is provided in the app to see what variances there are. Now all of this data now becomes available for use and analysis at the higher level, and we'll talk a little bit about that in a moment. We also see the emergence of other apps under which I would classify as disease tracking. So two of them, one used in South Korea, self-quarantine safety protection, which is a little bit more feature-rich in relation to alerts, for example, and our own Jamaican Jam COVID-19 app. And these, the common pattern in these apps are that they collect information on the user's health, they collect information on the user's locations and places they visit, and then they use GPS-based tracking information based on the user's devices. Of course, the user's devices are assumed to be on their person. They also provide alerts, all right? So for example, both Jam COVID and self-quarantine safety protection provide alerts that notify authorities whether an individual ordered to self-quarantine sneaks out. In the self-quarantine safety protection app, it provides alerts to citizens when they are found to have been in contact with a positive case. So the app has information about a person's test results. So if they have been positive and you have the app on your phone, then once you come within a particular distance of them, let's say close enough to them to be considered as outside of that social distance within which the disease is known to spread, then you are alerted that you have come in contact with somebody who has been tested and has been positive for coronavirus. And so the telephones actually communicate via short distance wireless communication in such cases. So this is really, really useful, and that can help to guide people as to how they need to respond having come in contact with someone. Of course, we know the disease takes a few days before we start having symptoms, and so you could be a carrier and not even realize. Okay, so once the data is collected, it is used in many different ways, and we have seen an emergence of many different applications, online tools, which I'll talk about. And of course, many researchers have used this data to do novel things. So here you see an image of healthmap.org, which actually records data in relation to positive cases across the world and provides color coding of where the disease has been spreading in terms of new cases, right? Cases below a particular threshold, and so on. So the data that is being collected by these apps and in general by institutions within the medical field is now made available for use in different ways. And so collected data is sometimes used for basic reporting. It's used for visualization, such as the one that you see here. And from the image, you will note that the colors indicate where the greatest number of new viruses, and that's in blue, are occurring over time, for example. Now, visualizations are very useful for policymakers, for people to see the impact or to be able to register what is happening and where. And this is an easy way to see what's going on across the world. And this is as a result of much of the data being collected by institutions, and in addition to these applications that have been built for mobile use. Of course, the information informs the public. It provides input to decision making. And of course, many policymakers have depended upon this for determining what orders, what policies, what new rules need to go in place in order to try and curtail or restrict movement and, by extension, the spread of the virus. Of course, we have other data sources. So, for example, in April 2020, Facebook and Carnegie Mellon University entered a partnership. And so what they've been able to do is to develop a specific application built on Facebook, right, which prompts users to take an optional survey. And the hope is that they would get millions of people to take that survey. All right. So their goal was to gather data at the country level on people who have COVID-19 symptoms. At the point when the partnership was being brokered, they didn't have good data regarding the symptomatic infections across the US. All right. So they saw the importance of gathering that data because it could be extremely valuable to help them to forecast spikes in symptomatic infections and also get some indication using the techniques that I've started to talk about of what spikes in hospitalizations may occur in the short term, for example, in specific areas. Now, they were also trying to get other big tech companies that collect data, which have large data sets, for example, to join in the project so that they could collect even more data, because the more data you have, the better you're able to apply some of the techniques that are covered under machine learning and forecasting. So I've just talked a little bit about the data sources. We see that sources include, for example, the medical institutions that record test results and other data related to the disease. We see apps that record data from individuals. And so there's a kind of crowdsourcing of data. And then we see the big techs that have implemented technologies on top of some of the popularly used programs in order to also collect data. And so we now have a lot of data coming in. So it's now time to talk a little bit about what kinds of processing have been done on this data. And this is the part that we usually get excited about, because here is where we discover patterns. For example, we're able to use the statistics or statistical techniques to build models, for example, and help decision makers to get a sense of what the future holds, whether it be short term and long term. And so I'll talk a little bit about some of what has been done with this data. So for example, Alquan has developed an interactive tool that uses some differential equations to try and model the projected numbers of infections across US states. So here they are trying to predict down to maybe 3 months, right? Based on current numbers, death tolls, reported infections, right? What is the number likely to be in different US states over a particular period? And in this model that is visualized here, they have from June to November. And so it's kind of giving a sense of what is likely to happen. Okay? So that's one long-term infection prediction model. And this depends upon a lot of mathematics, of course, with differential equations going in with the current data. We also have some statistical machine learning that have been applied. So here, this model comes from Columbia University. And Columbia University actually produce inference projections and simulations of the impact of interventions. Okay? So what is likely to be the impact of having a lockdown, for example, in this city or in this county over this period of time, for example? And they provide that information to decision makers. And of course, they're using statistical machine learning techniques. Now, one of the things they've been able to do is to estimate the infection fatality risk in New York from March 1 to 16 as a kind of short-term model for the decision makers to see what is likely to happen. They've been able to build a machine learning model which shows associations between the built environment, the neighborhood socioeconomic status, and the infection among pregnant women also in New York. And here I have a link that you can check out to get some more information about their model. And as you would see in the diagram here, they have used what we call training data to train the model. So that's some background data. And then they have tested the model using a subset of that same data. So for example, typically what we do is break the data like in 80-20 or 70-30. We use 70% of the data to train a model. And when we get that model, we use that next 30% that was not used to train the model in order to test. And then we measure the accuracy of the model operating on that 30% or that 20%. And here they produce different scenarios, right? And so we see several lines showing what is likely to happen in scenario 1, scenario 2, scenario 3, and so on, in terms of the number of daily new cases, all right? Here we have another application of machine learning by Gunjan Hung of Brown University. And here he uses human encounters and the density associated with that to predict reproductive number and hence daily and weekly new cases. And so in his model, he produces a confidence interval, right? He uses data that is updated periodically. And typically when I look on his model, around about once every 5 days to a week, new data is passed into his model to kind of predict in a kind of short term. And so his modeling is short-term prediction, unlike the other models that I've talked about. Here, he's trying to predict on a weekly basis what is likely to happen. And so, as a matter of fact, he was able to predict spikes in cases in the US correctly by using his machine learning model. And here's a link to the article that speaks to that actual prediction on a particular week. Of course, we have seen where the data has been used to generate simulations, and there's a team of researchers from Georgia Institute of Technology, Georgia Tech, Value Analytics Lab, Boston University, and Harvard that have now started to use simulation to try and show what is likely to happen depending on different interventions. And so different interventions may result in different scenarios. And so there's an interactive tool that you can find at the link here. It's called a COVID-19 simulator, where you can pass in information about different things that are done in order to curtail the COVID-19 spread. So for example, if there's a partial lockdown, if there's a curfew order, if there's restricted movement, and so on and so forth. Now, what they have done is to build a simulator which applies some techniques in order to model the likelihood of changes in the new number of COVID cases each day resulting from these measures being applied. And so what this is able to do is to simulate what is likely to happen if it is that the government or the local authorities decide on a particular thing versus another thing. Okay? And of course, you can go there and use the simulator. It is accessible. Data scientists have also tried to predict the life of COVID-19. Is it here to stay? When will it end? Will it end? What is likely to happen? And so, two researchers, Hussein and Khan, published a recent paper called A Predictive Model for COVID-19 Spread Applied to 6 US states. And they made some bold predictions. First thing, they suggest is that in continued lockdown, COVID-19 infections will persist for at least 2 years. They also predict that infections, however, will rapidly rise by an order of magnitude if lockdown is lifted, peaking after 2 months, recurring yearly similar to influenza. And of course, they are using data to build models in order to draw these conclusions. They also predict that repeated lockdowns and releases do not avoid an endemic infection, even following a White House release guideline of 14 days of decline in cases or even a stricter 28 days. They further note or are predicting that earlier lockdowns would have cut COVID-19 deaths in New York, but surprisingly, not in Texas. And so they have found that there are differences that are contextual, that are specific to the local domain. And one of the final things that they said is that infections could be curtailed by stringent measures like strict lockdown compliance, face masks, social distancing, contact tracing, and isolation. Now, from these predictions, based on their modeling in that paper that they published, we recognize that even from point 2 that I have here, that the notion that infections will recur yearly, similar to influenza, and if they are right, then COVID is really here to stay. How do we manage? How do we curtail? How do we restrict? I sincerely believe that continuing to collect the data, using these techniques to do modeling, simulations, and advise the policymakers as we move forward is critical. And so data science is critical to the management of the COVID-19 pandemic. Thank you for your time and attention.
Thank you so much, Dr. Anderson. We have witnessed the power of data-driven decisions here in Jamaica in the fight against COVID-19. And your presentation has certainly shown us the wider impact of data science as a powerful multidisciplinary tool in managing and indeed fighting COVID-19. Our next contributor is Professor Rupika Delgoda. Professor Delgoda is the current director of the Natural Products Institute, NPI, here in the FST at the UWI Mona campus. She holds a Doctor of Philosophy in Pharmacology from the University of Oxford, a postdoctoral research fellowship in biochemistry, and a BSc in chemistry from the University of Papua New Guinea. Professor Delgoda's research really touches the heart of Jamaican culture and focuses on exploring and unveiling the biological activity of Jamaica's natural resources. So she assesses the efficacy of medicinal plants, yes, Jamaicans, bush medicine. She evaluates ethnomedicine, which really means she examines health-related knowledge that people inherit from their ancestors, which are based on bioactive compounds in plants and animals. She also assesses the safety of medicinal plants, and this is crucial if we are to derive the full benefits from our flora and fauna. I know all Jamaicans, both here and abroad, will enjoy Professor Delgoda's talk on the potential of Jamaica's local natural products, nature's gifts, opportunities, and challenges. Please help me make her welcome.
Thank you, Dr. Singh-Wilmot, for that introduction. Thank you also to the organizers of this forum for providing this opportunity for me to present some research that's coming out from the Natural Products Institute. I'd like to speak to you about nature's gifts, the opportunities and challenges. Jamaica is blessed to sit in a biological hotspot. It has varied species of both terrestrial and marine organisms, and a third of its higher vascular plants are only found in Jamaica. That's 28% of endemism among the higher vascular plants. In addition, our own research shows that there is a traditional and cultural knowledge system that is mingled with those hailing from Africa, Asia, and Europe, mixed with a Jamaican own ingredient that makes a traditional knowledge system here that hails from centuries. Our research from the Natural Products Institute shows that 73% of Jamaicans self-medicate. That is reported to be one of the first quantitative estimates in the world, following the guess estimate made by the WHO that 80% of developing world rely on natural and traditional medicines to heal. This is important because, as the graph represents, if you notice the traditional medicines that are used to test for inventions of drugs, the chance of discovering a hit or a new drug is much higher coming from ethnomedical practices rather than from combinatorial chemistry, molecules that are randomly picked for such searchers. Obtaining drugs from nature is not uncommon. Man has been searching for solutions from nature for centuries, and indeed pharmaceutical drugs have been discovered from nature. Mean contribution of natural products, small molecules, over 39 years indicates that 32% of those have been either directly derived from natural products, their derivatives, or inspired by them. The largest contribution for drugs have come for antibacterial solutions or anti-cancer drugs. And as you can notice, 14% of antivirals have been inspired by nature. A majority of antivirals have been in the form of vaccines. But you would notice that vincristin, vinblastin came from periwinkle plant, Taxol that came from the Pacific yew is a drug for cancer. And of course, our own Canasol that came from cannabis plant. Let's pause to take a look at nature's expressions. You'd notice that there are hundreds of chemicals expressed in one plant. Mother Nature expresses small molecules known as secondary metabolites. They're known as secondary metabolites because they are not necessary for the primary function of a plant of breathing and living. However, these secondary metabolites are key for an adaptive advantage and for the evolution of plants. Arising from a single cellular plant organism existing in a soup of harsh chemicals, it has evolved to cross over and become a terrestrial plant, a multicellular organism. At each stage, it has relied on secondary metabolites for granting it an adaptive response or an adaptive advantage, be it to help it avoid desiccation, be it to prevent itself from getting a fungal disease, be it to prevent itself from getting overgrazed, or to provide it a reproductive advantage by producing beautiful colors and fragrances. So these advantages have come from, as I said, small molecules called secondary metabolites, and there are hundreds of them. What I want you to also notice is that their expressions can vary not only from plant to plant or plant species to plant species, but also from one plant to another, and depending on the soil conditions and environmental conditions, as well as expressions varying from seasonality and soil conditions. Our surveys have indicated that Jamaicans prepare their ethnomedical preparations from all plant parts. That's leaf all the way down to the stem, the branch, the seeds, the roots. All parts of the plant has been used in ethnomedical preparations. And Jamaicans also prepare their ethnomedical treatments using different preparation methods. That's decoctions, where you put all your ingredients together and boil it for a long time, to infusions like a tea, to tinctures, where you are actually placing it in alcohol for a number of days. And again, what I'd like you to notice is that we have a number of different ways of preparing them, a number of different plants that you take to make such preparations, all of them leading to the point that one has to be aware that the chemical constituent that you are extracting or getting out of the plant can vary significantly depending on the part that you use and how it is prepared. And therein lies the value of ethnomedical preparations. That recipe is key. Our research shows that Jamaicans self-medicate for multiple purposes, for multiple ailments. And you'll notice that the highest percentage is used towards respiratory systems. This slide also shows you the top most commonly used herbs for the treatment of cold and flu symptoms. It's led by leaf of life, or Bryophyllum pinnatum, and it has garlic, susumba, rice and peas bush, and so on. So let's shift gear to something that is current. The severe acute respiratory syndrome coronavirus 2 is a positive strand RNA virus. The virus is coated by spike proteins. It has an envelope of glycoproteins that has these spikes that are very critical to recognition of the site of binding as well as adhesion. Once it enters through the throat and tongue and nasal area, it will then take resident and move to the epithelial cells of the lungs, where most of the damage will take place. Now, what we want to be able to understand is is there any way in its introduction, in its adhesion, and its multiplication, whether there are any ways that we can find to intervene in that process. So, virus entry and binding to the ACE2 receptor is key. From there, multiplication of the virus takes place, and then on to the third and the fourth stages of the disease, where it increases, the symptom severity begins to increase, where white blood cells count goes up, the platelet and lymphocyte counts decrease, and very importantly, as the severity increases, the systemic pro-inflammatory cytokines and biomarkers such as the interleukin-6 and interleukin-10 and the tumor necrosis factor increases. Meta-studies have shown that these are the markers that physicians need to look out for as the severity of the disease progresses towards multi-organ failure and then sometimes towards death. These multi-symptoms then allow us to think about the multi-targets. How is there a way to have prevention of binding? Could we have non-specific protein binders in the viral envelope? Can we think about ways to prevent that virus from replicating? And are there ways of interventions for reduction of the severity of the disease, where you can have anti-inflammatories and the decreases in the biomarkers that we had spoken about? Towards that goal, we decided to look not only at the top 10 herbs that Jamaicans use for self-medication purposes. We took another step back and decided, let's look at the natural products that we have investigated for other purposes throughout the last 15 years or so that we've been doing research at the Natural Products Institute. And this has been plants with antioxidant activities, anti-cancer activities. These have been from ethnomedical as well as endemic plants. So what I did was to sort of give you a quick overview of a few plants that we have looked at. And I then want to highlight, even though this is a busy slide, I want to highlight a few of the phytochemicals that are present in some of these plants that might be very interesting towards the targets that we had identified earlier. So you'd notice that a number of these plants, in particular preparations of infusions, express something called tannins. Tannins are the ones that are responsible for the tea having some sort of a very sharp taste and an aftertaste that leaves in your mouth following the consumption of the tea. These tannins have been shown, they are actual non-specific protein binders. Therefore, they've been shown to bind with the viral envelope that enters through the mouth, and that binding then prevents the viral envelope from adhesion. This has been shown particularly for highly concentrated amounts of procyanidin B and towards influenza virus. Gaining evidence from that, some authors believe that preparations of highly concentrated tannins through chewing gums, through lozenges, through mouthwashes, may actually help to prevent the viral envelope from adhesion at the point of entry. You'll notice that this will be a temporary prevention and therefore not a drug and not a solution or an offer that can be used for permanent treatment. But certainly in terms of ethnomedical preparations, this is something that we could think about because the number of tannins that are present in all plants that we have investigated is quite high and prevalent. You'll also notice that the bolded forms of certain plants that we have investigated ourselves for anti-cancer activity shows that there is a reduction in the interleukins 6 and 10. For example, jackfruit, we were able to show that through an animal model that those values, anti-inflammatory properties, are high and that the TNF-alpha and the reduction in the interleukins can go down with specified standardized preparations of this plant. And so for the bastard cedar and Gossypium lifolia, you'll also notice that these are so for guinea hen weed and the leaf of life that we had identified as the top most commonly used plant in Jamaica. So this means that there are interesting phytochemicals that are expressed in Jamaican plants. It also means that there are mixtures that expresses them in the way the ethnomedical preparations are conducted. However, it does not necessarily mean that we could make that leap to say that drugs can be made from it without the proper procedure being followed. So a pharmaceutical path is possible as long as those phytochemicals can be isolated and their studies are to understand their efficacy in a human model with all the safety profiles to go with it is a possibility, but it is a long road. What we'd like to do is more to look at the ethnomedical preparations, which is a mixture of phytochemicals that we know exist together. But what are the challenges of working with such mixtures? Well, challenges and opportunities reside side by side. So I'd like to take an example of an infusion that we had examined in the laboratory, Hyptis verticulata. This is John Charles tea. And we were examining it for a particular bioactivity. In this case, it was an enzyme that we were interested in, cytochrome P450 enzymes, and we were observing its inhibitory patterns. This particular graph represents the inhibition, that is the concentration that is required of either the tea or the active ingredient we are looking at, the concentration that is required to obtain inhibition. Therefore, if it is very potent, the value would be low. And you'll notice from this graph that the mixture or the extract has a very low value. In other words, you'll require very little from the extract to obtain inhibition. And all the other compounds beside it are phytochemicals. These are single ingredients that are found in the tea. They are phytochemicals or secondary metabolites found in the plant. And we were then able to examine each of these ingredients independently. They're single, purified phytochemicals. We examined its inhibitory components individually. So as you can see from the graph, none of the single ingredients were as potent as the mixture itself. And this is what we call synergy. In other words, when you put multi-components together, they act in a way that is far more enhanced than each active ingredient by themselves. So is it possible then to examine this in terms of a virus? Could we see synergy as opposed to additive effects? That means if you put number of single ingredients together, or antagonistic effects, that means that they nullify each other's activities. Could we observe that when number of components are put together, that the synergy is better and that we get better biological activity? Well, we then decided that we would subject some of our natural mixtures to see whether antiviral properties could be enhanced in a SARS model. That is to exploit the complexity of natural components as antiviral monotherapies or synergies together with FDA-approved antivirals. Can we see an enhancement of the antiviral properties of an FDA-approved drug when Jamaican mixture can be in the presence of a Jamaican mixture? So the questions to answer are, can we evaluate these monotherapies? Can we evaluate the value of the synergist that enhances the monotherapies? And then identify the impact of the natural compounds on metabolism, mode of action, and then also understand the impact on the microbiome microbiome impact on the drug mode of action. So these are the questions that we'd like to answer, and we're working in collaboration with the Colorado State University to get these answers done. And so again, what we plan to monitor are 3 steps. Could we observe, as the concentration of the natural product increases, can we observe whether the gene replication goes down? We would be identifying a particular gene. Is that particular gene identified from the virus? Is gene replication impacted in the presence of the natural products? Can we see cell-to-cell spread using a particular fluorescent probe that we would be inserting in the virus? Are we able to observe whether the virus is actually replicating? Is it spreading? Can we then also see the virus output by conducting a plaque assay? These would be conducted in non-human primate vero cells and that have been infected with the SARS virus. There's some precedent to experiments that we plan to do with the SARS virus because we have examined plant extracts in the past, Jamaican plant extracts, against the chikungunya virus. What you see from this slide is a plant extract from Jamaica that we had investigated on the chikungunya virus that were co-infected again on vero cells. The left-hand side shows the fluorescence, that is a thriving population of viruses on the left-hand side when there is no natural product present. And as you slowly increase the volume and the concentration of the natural products, as we go across to the right, you will notice that the fluorescence decreases. This decreases because the number of viruses that can exist goes down, and therefore it shows that the chikungunya virus has been impacted, its viral replication has been severely impacted in the presence of the Jamaican natural product mixture. So we hoped that we would be able to conduct similar experiments with the SARS virus. I'm going to change gears somewhat to talk a little bit about another area of research that we constantly think about when developing therapies from natural products, and that's to take a look at safety. We have conducted 4 surveys throughout Jamaica that have looked at, we know that Jamaicans self-medicate with herbs, but what about the population that's on prescription medicines? We conducted 4 surveys starting from 743 patients who were patrons at pharmacies and asked them the question, are they self-medicating with herbal remedies while they are on prescription medicines? So these 4 surveys amongst cancer patients, amongst pharmacy patrons, as well as house to house, allowed us to notice that the answer to the question, do you take botanical preparations along with prescription medicines, is overwhelmingly yes. 81% of Jamaican adults and 76% children were self-medicating and using botanical preparations while they were on prescription medicines. Alarmingly, 80% of physicians are unaware of such co-medication practices. So just to take another step back quickly to understand why is that important. Well, the chance of interactions of a prescription medicine along with a natural product can be clinically relevant and can therefore lead to adversities. The reason why that happens is that a majority of adverse drug reactions arise from a pharmacokinetic interactions coming out of an enzyme system called the cytochrome P450s. These enzymes are responsible for the metabolism of the prescription drug to its often harmless metabolite, which is cleared away out of the body. If the cytochrome P450 enzymes are interfered with in any way, if they are inhibited or if they are induced in any way, then, as you can imagine, the clearance of the drug can be impacted. There are numerous drugs. There are over 70% of prescription medicines that are reliant on cytochrome P450 enzymes for their metabolism. And therefore, any medication or natural products that would interfere with the functions of the cytochrome P450 enzymes could have potential interactions, some that may be clinically relevant. And how is that so? If you have a natural product that is an inhibitor of cytochrome P450s, then the co-medicated prescription medicine levels in the blood could increase, leading to toxicities. If the natural product that you are taking is an inducer of this enzyme, then it means that the prescription drug that you're taking could be cleared away faster, leading to subtherapy or absence of therapy. And this is the reason why we have been studying drug-herb interactions in a country where a majority of people are self-medicating and physicians are unaware. Drug interactions are particularly important for those with comorbidities at this particular time. Those with hypertension, diabetes. We all know that persons with underlying conditions are at higher risk during COVID period. And what I'd like to make a note from this slide is that many medications, particularly for hypertension, they're all subject to metabolism through the cytochrome P450s. Such medications, then, if you are going to be using together with any natural products, one has to be aware that drug interactions is a possibility, and the note I would make at this stage is to talk to your doctors about the potential for such interactions. we took the most popular types of teas that people co-medicate and conducted in vitro evaluations of these teas on the cytochrome P450 enzymes. And we recognize that their in vitro means that the clinical relevance is yet to be conducted, but we are able to shortlist the plants that we would like to work with. That these are guava tea leaf, search me heart, ackee tea leaf, again, John Charles tea, and so on. So now that we have shortlisted them, we are poised to be able to take another look to evaluate clinical relevance. These things have been collated in a publication called Potential Drug Interactions that are available for physicians and pharmacists. So how is that relevant, again, returning back to the botanicals that we would be considering for COVID development? It's just a caution to notice that the leading drugs that are currently being used, remdesivir, dexamethasone, azithromycin, hydroxychloroquine, some of these are subject to metabolism by the cytochrome P450 enzymes. And similar to hydroxychloroquine, again, because it's subject to CYP3A4, there could be interactions with John Charles tea, grapefruit juice, and St. John's wort. St. John's wort is an inducer of CYP3A4, and if somebody is self-medicating using St. John's wort, then the likelihood is that you could end up with subtherapy because the drugs are cleared away faster. So in conclusion, I'd like to point out that Jamaican botanical preparations as used in ethnomedicines contain multiple active ingredients. Many of the plant preparations contain valuable phytochemicals useful, that could be useful to fight viral binding, replication, and symptoms. The mixture of chemicals could have synergistic effects useful together with multiple targets, and that the botanical preparations need to be well characterized for the quantity of such chemicals that we call standardizations before any such claims could be made. And the mixtures need to be studied using useful models, but we also look forward to results on the COVID replication experiments. I'd like to end by thanking some of my collaborators who I have worked over a number of years, particularly the faculty members from the Faculty of Science and Technology, from the Department of Chemistry, who have been working with us for isolation, characterization of natural products, as well as others who overseas, as well as local, who have helped us to look at the biological activities of Jamaican natural products. Special thanks to Dr. Pereira from the Colorado State University and her team, who is working in collaboration with us for the COVID assays. Thank you all.
I join with you, the audience, here in Jamaica and in the world, to thank Professor Delgoda. Jamaica is blessed with valuable resources in the fight against COVID-19. Now, if these chemicals are weapons in this war, if these natural products can be weapons in this war, then Jamaica is ready for the fight. And we are grateful to have scientists like you, Professor Delgoda, and your team at NPI, leading this fight, leading this way, and guiding this process to harness traditional knowledge and couple it with high science, so that we can derive the benefits of it and the benefits of our natural products while protecting our population. What we really need now are partnerships, you will agree, to do more research and to translate this research, to translate what we already know into commercial activity. Now, later on in this forum, we are going to talk about some of those partnerships. We're going to talk about how valuable they are and how they can work. But right now, I want to arm you with some more information that you can use to protect yourselves against COVID-19. So now we go to our section on tackling COVID-19 misconceptions. And we turn our attention to another of the UII's outstanding women in science. In fact, this female, Dr. Sylvia Mitchell, was recently named as one of the 70+ UII women of distinction. Dr. Mitchell is a senior lecturer here in FST at the Biotechnology Center. She heads the Medicinal Plant Biotechnology Research Group, which she initiated in 1999. Before that, Dr. Mitchell worked at the Scientific Research Council and the Sugar Industry Research Institute. She holds a double major BSc in Geography and Botany and a PhD in Biotechnology. She is trained in various aspects of technology commercialization and has over 34 years of experience in plant tissue culture. Her area of research is developing medicinal biotechnology solutions related to plant tissue culture using locally grown plants. However, ladies and gentlemen, today, Dr. Sylvia Mitchell has sort of shifted her focus, and she is merging science with common sense in a talk entitled Size Matters, the Science and Common Sense of Wearing Masks. Ladies and gentlemen, I recommend that we all lend our ears to Dr. Sylvia Mitchell.
Thank you, Dr. Singh-Wilmot. Thank you, FSD, for this opportunity to share on this subject. This is based primarily on a Gittena article that was put out in May 16, 2020. We want to share today about face masks. The some sense, there's a lot more, but we'll just share some, and the common sense coming out of that science to inform what matters and what doesn't matter. So, let us set the scene. I'm going to share from two areas of my work. One is on ethnomedicine, and we're going to be looking at one aspect, which is prevention better than cure, and as a biotechnologist working with microbes. We're going to look at size, the science behind mask wearing, and common sense. This is on a background that this SARS-CoV-2 virus is highly transmissible. It's a basic reproduction number of 2.4. That means each person that has it usually infects at least 2.4 other people. Take in mind that a lot of those that have the virus are asymptomatic, which means you can't really tell that they have it. And before you have symptoms, you can also transmit. Period range from 2 to 15 days, medium length 5.1 days. So at that stage, in those initial days, are actually when the viral titer is the largest, whether you are asymptomatic or presymptomatic or symptomatic. And these particles have been found in saliva, nasal secretion, and in tears. Yes, your eye. So we're going to look first at this idea of prevention being better than cure. It's better to stop something bad from happening than it is to deal with it after it has happened. So we're going to see how can we prevent in the first place getting this virus at all. And we are looking then at our folk medicine, and notice that there are many examples of preventative aspects of our folk use. One of them is that we do tend to be a very cleanly people. We clean ourselves, we bathe often, we wash our dishes, we are always cleaning, and that's a very good aspect. And it has resulted in one thing that we've seen, that communities don't have to be told. They cordon off their community. Anybody who comes in there, there's a cleaning station, and they are in control of their space, which is very good. The mothers have a tendency of the children going back to school, giving them a washout, removing lice, and giving worm medicine, whether or not the child is sick or not. Root tonics, mainly for the men, for strength and vitality. Raw foods, use of fruit and vegetables, herbs and spices. Nutraceuticals, these are being used now as immune boosters. And medicinals, we have a lot of cold and flu medicine. Nobody has to tell them to take a cup of tea. We also see it in the application of scientific research. For example, guinea hen weed has got a lot of news as an anti-cancer, you'll find that it's being used by the folk now and again as a preventative. So let's explore how we can prevent COVID-19. The other aspect is, as a biotechnologist, we measure things in centimeter, which is about that big, and millimeter, which is about that big, but we are more comfortable in the area of microns and nanometers. So a micron is about the size of a gene, a piece of DNA. It's a bit smaller than a bacteria. And then the COVID virus is about 0.125 microns wide. If you look at the top, that one shows if you had enlarged a human hair on the left of that top implant at the right, then that dot beside it would be the virus. So you have the human hair, fungus, bacterium, and that little dot, the virus. So you can see it's not invisible, but it is not visible to the naked eye. Bearing in mind that a micron is 1,000th of a millimeter. Okay? And you can also see some comparisons there, fungi, yeast, and bacteria. In the plant lab, we have viruses too. They grow in the plant, but they do not get up into the meristem, so you can take it off, you can get a virus-free plant. You grow it on a media, just one bacteria or fungal spore on that media, and it overgrows the whole media. So the microbes tend to grow very fast. So what we want to do is stop that first one from landing. We use something called a HEPA filter, which is on the bottom right-hand side. And the HEPA filter, there's one at the top filter that filters out the dust, and then we have a HEPA filter at the back, which takes out 99.97% of particles that are smaller than, or bigger than, 0.3 microns. Okay? So it will take out all our bacteria, our viruses, and the fungi. So we can work in there. But we've also designed at the top right something called an MMU. And you can there exclude microbes. You wipe down the inside with alcohol, and you can work in there and again get the area free of fungi, bacteria, and viruses. I don't know if you've seen the shows where they put up this plastic and then they operate inside of it. This is the same principle. So how do we then make our own personal mobile unit that can go with us and give us a space in which there are no microbes? And this is the idea behind the personal protective equipment. Some lessons, practical lessons learned that are relevant. The viruses do not multiply on their own if they are not in a host. So once they're floating around in the air or landing on a mask, you don't get this rapid multiplication. That only happens within a cell. So the same number of particles will be on a hand until you wash it off. Just remember, no touching of the eye, nose, or mouth. That is one advantage of wearing the mask. It encourages you not to touch sensitive areas, whether or not there is a problem. It also means, though, that when you get inside, you need to sterilize your hand before touching doorknobs, keyboards, telephones, etc. The biggest arsenal we have against this virus actually is soap. And that is because one side of the soap, see the picture in the middle there that looks like a tail, is oil soluble. And it gets into the oil, that outer oil, lipid layer of the virus, and it actually causes it to break apart. Once that coat is broken apart, the inside, which carries the information for it to make more copies of itself, cannot live, and it is no longer infective. And this is why you are told to wash your hands for 20 seconds. because it takes that amount of time for the soap to actually do its work. Alcohol works similarly on the this outer lipid layer, but it does dry off the hands. So soap is preferred, but if you don't have soap, you use your alcohol. And of course, for surfaces then, the soap or the alcohol can work, or bleach is also good, but don't mix them. So we're going to look now at size matters. As we said, this droplet, this virus is living in, is transporting in a little layer of water, and as it dries out, eventually it will no longer be infective. Again, at the bottom left-hand side, you see the little droplet that would contain the virus, and you see the size of a human hair. So just imagine one of your hairs, and you blow it up to that level. That is how small one of those virus particles is. It's about the same size as the influenza virus and the SARS virus, which is again on the bottom right. So, what are the conditions under which we will encounter, or that people encounter these virus particles? Again, we look at size. So when you talk, when you cough, when you sneeze, then different size particles come out. When you cough or you sneeze or you sing, the particles get to go further away, but when you talk, they tend to be more smaller particles that are called aerosol. And the point here being is that the bigger the particle is, the more tendency of gravity to pull it down. So if you talk directly to someone, that's a problem, or however close you are, if you're shaking hands, or if it drops on a table. So here's a little pet peeve that I have, where you're told to sneeze into your elbow, and then you're told to touch somebody with your elbow as introduction, or to see. And I see that as a no-no. Because if you sneeze into your elbow, it means your elbow now has a possibility of having all these virus particles. And it's best in this situation to just think of everybody having it, right? So you start seeing things at a level below your eyesight, because you're looking at where is your breath going, where's your sneeze going, where's your tears going. And if you're going to sneeze into your elbow, you do not then want to be giving it to somebody else. Use your foot instead, or just say hi, all right? And not, hey, just a hi is fine. Another aspect of size is the poor size of the masks that are being put out. Those 9 squares, if you look at the third column, which says surgical mask, it has 3 layers, and you can see that the different fibers are interwoven. And that means it is going to be a little more difficult for the viruses to get through. If you look at the cloth mask on the left-hand side, you're seeing those pore sizes, and they're between 80 and 500 microns. So this is one study. And if you look down to particulate matter like smoke, which is 10 microns, which is an order of magnitude from this virus, but for our sake, looking at the mass, that was filtering out 63 to 84%. So even at 10 microns, it was too small, and it was getting through those masks. And if you look at the masks, you can see they really look flimsy. And it is a credit to the Jamaican, as again, having an idea of this cleanliness, that when they make the masks locally, they're not flimsy, right? They have 2 or 3 layers. So this would be, I would say, like the least mask that you would wear. Another aspect that you have to think about, we're told to wash them, is that when you wash them, the holes get bigger. And efficiency drops, in this case, after the 4th washing and drying cycle. So it's best when you get them home, you put it in soapy water. Don't, you know, we love to squish, 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 squish. Don't squish, squish. Just leave it in the soapy water and then rinse it off with water and hang it up and let it dry. Okay? So, what does this N95 mean? N95 means it removes 95% of airborne particles that are 0.3 microns or bigger. Notice ours is 0.125, and that 95% means that you're losing 5%. 5% is getting through. Actually, N100 means none of them will get through, at least 99.97, but those are not around. What is helpful with the science, the masks that we do use, is to use different types of material. So they found cotton with silk or chiffon, and definitely at least 3 layers. What you're doing is trying to reduce the risk. You won't remove it completely. So this one on the left was showing a comparison between these homemade masks and a surgical mask. Not very bad at all, much better than wearing no mask. And tea cloth, which tends to have a closer weave, was actually getting up to the surgical mask, and then you have another to the right, the FFP2, and those on the right were looking at smaller particles around the range of the COVID particles. This one was done on the left with an influenza virus, as I said, similar size, and a scarf was basically no use at all, okay? So when you have a scarf and you put it over your face, you're only trapping about 1% of anything that might be out there. You're giving yourself a false sense of security. And as you go down, I'll get better. You have scarf, 100% cotton shirt, silk, pillowcase, linen, cotton minks, and tea towel, with tea towel being the better of those set. And then vacuum cleaner bag is actually a HEPA filter. So if you can use that as the inner portion of the mask, that would give very good support. Another study on the right you can see is by pushing the mask to have a closer fit, you are actually able to prevent more particles from entering or leaving, depending on what is the issue, whether you have it or you're trying to prevent yourself getting it. So the science behind mask wearing concludes, and there's lots of things on this slide, basically showing you the number of particles in a cough or a sneeze, that there's a lot more in a sneeze, that there's a half-life of those smaller aerosol droplets of about an hour. So somebody could have come into an area and have left already, and there's still enough particles in there for somebody coming in to be affected. But the science so far, and you know science has to do a lot of replicates and experiments, but each of them is basically saying the same thing. Having a mask on is better than not having one on at all. And having a mask on a person who has it and the person who's trying to prevent getting it is the best situation. And as I said, depending on the type of mask, then gives you even better advantage, and the fit of the mask gives you another advantage. So we're going to look at some common sense interpretation of this science. And this is a nice little graph here, and there's lots of them now in the literature, but basically what it's saying is that first of all, going across, we want to get from the yellow to the blue, is you increase the efficacy, that means you're blocking more of those virus particles going through. You can get to 100%, But if nobody's wearing the mask, it doesn't make no sense. So we also need, and that's the one on going up on the left, adherence. We want most of the people to be wearing the mask. What we want to get to is from that 2.4, one person infecting 2.4 people, to one person infecting less than one person, and then the virus is going to peter out and will not be found in the population again. So, common sense. 2 reasons for wearing a mask. First, you want to protect, I want to protect myself from getting infected, so I'm going to wear a mask. It needs to fit snugly. They need to, especially around the nose. So this is one I would wear if I need to go in somewhere fast. It fits here quite well. It fits here quite well, but it could be tighter. See, like that, it's tighter. But I still have a space on either side of my nose. Now, depending on the shape of your nose, mine is quite sticky out. There'll be more or less space right there. So I have another mask, and what I've done with this one is give myself a little extra piece inside. And that goes on either, because I don't have a metal strip, that goes on either side of my nose, and now when I tie it, that space is no longer there, right? And as we said, the bigger ones tend to go down, the smaller ones tend to eddy, so you want to keep the front, the top secure, the sides secure, not over your nose, your ears, and then you want to cover your chin as well. So your mask really has to be big enough for your face. The small little ones and the big little ones, you need to think a little more about how snugly it fits to you. To protect others, if you are infected, well, if you know you have symptoms, you really should not be going out. You need to inform the authorities and self-isolate. The issue, though, is that you might have it and you don't know. So, if you wear the mask, you are still protecting others. What mask to wear? Well, the mask with the larger pore size is going to require the wearer to maintain a greater physical distance than one wearing a mask with a smaller pore size. And you notice it's a physical distance. We can still socialize. We have so many methods. We can WhatsApp, we can call, we can Zoom, so we can keep socially close, but we want to keep a physical distance so that those particles, if they are there, they are not going to get to us. So if everybody in a supermarket is wearing masks and you're not talking, you get what you want and you're going out, there's a much less possibility of getting any of the virus, if it's there, than if you were inside for 2-3 hours singing your heart out. Okay? So where you're going is also important as to what mask you're going to wear. Yes, it would be nice to use a HEPA filter. Yes, it would be nice to have an N100 mask or even an N95 mask. So if you have it, good. If you can wear it and keep it clean in between uses, good. If you have a surgical mask, that is the next one that is good. But if you do not, then a good cloth mask with 3 layers with this different type of material will work. But a cloth handkerchief, only if you have nothing else. So use the highest grade of mask that is possible. When are you to wear a mask? There's no scientific evidence that suggests that a mask alone is enough to stop transmission of the virus particles. In hospitals, masks are worn with a face visor, okay? Because it can also get into the eyes, so the face visor helps, or, that's the top one, or a full hazmat suit, which is a lower one, would be worn. Once I have that complete covering with the the plastic or the glass covering, then you need the oxygen. But if you have a face visor and a face mask, then you wouldn't need the oxygen. What we're aiming for is to reduce the risk of getting the virus. The 3 identified routes, as we said, are the saliva, the mouth, nasal secretion, the nose, and tears from the eyes. So we wear a mask, especially where the risk is greatest. Here you assume everybody has the virus. However, when leaving that high-risk area, how you take off the mask, store it, wash your hands and other parts of your body where the virus might have landed before touching different areas, including your eyes, nose, mouth, or knobs or phone, is highly critical. So even when you have on these suits that totally protect you, you are finding some nurses and doctors who are getting infected because it's how you take them off and how clean they were before you put them on is highly critical. So, this very quickly, some common sense of all of the data that is out there. How do you ensure your fabric is a good filter? Hold it up to light. The more light blocked by the fabric, the better it is. So if you can see the light coming through, that one is no use. Okay? Another one is if you have a lighter flame and you shouldn't be able to blow it out. They should be of good fit, as I said. You can make your own mask with a combination of material. So I talked about the sneezing in the elbow. The other thing is if you accept change in the bus, the virus can be on the money. So accept it into a plastic bag, spray inside the plastic bag, leave it for a time before you add it to your purse. Prevention is better than cure. Little bit of work make for a lot of saving. How to wear the mask? Well, you have to figure which way is up. Those that have the metal strip is at the top. As I said, it is there to hold it against your nose, to take out that little area right there. If not, you have to find another way to close the nose. And as I showed you, this is how I used my little thing to do it. Make sure it's snug. Hey, I see a lot of people. They take off the mask, the top. Some of them leave it so. Some leave it so. Some leave it so. All of them wrong, right? What you need to do, take it off, put it straight into a plastic bag, clean off your hands, talk. When you're finished, everything there needs to be sterilized. You take out a new, different mask and put it on. Otherwise than that, you keep on the same mask and talk through the mask. Problem is, if you are infective, if you take off that first mask, you will infect people. So it's better to keep it on. So I talk a lot about going out and having more than one mask, use the highest quality, stay out as short a time as possible, avoid crowds. If you want to take off the mask, don't. Keep it on even when talking. If you do take it off, put it in a separate plastic bag. Afterwards, put on a fresh mask. Wash hands with soap and water before moving the new mask from its container. So when you're coming in, take off the mask without shaking it. Dispose it straight into a garbage bag or into a bucket with soapy water at the entrance of your house. Wash your hands and face with soap and water at the entrance. Then you go straight to your bathroom and bathe. Reusable mask, after washing and drying, store it in a plastic bag until you use it. 1 mask per bag. Do not reuse cloth masks if they get stretched. So, I hope that you have been able to learn something. I have learned a lot from this experience of COVID-19. I have really learned that we do have an ethnomedicine principle of prevention being better than cure. If we can prevent, and we have been doing an excellent work with prevention, then we will not have to fight with all the ways of having to cure and the possibility of death. Sterilization practices of biotechnologies can help us to decrease risk during this time. The judicious use of face masks still is not enough alone. It has to be supplemented with hygiene, washing of hands, especially soap, alcohol, washing down areas with bleach. Indoors, you have to watch the ventilation, the ACs, avoid crowds, keep your physical distancing and all the other measures that we have been using, like contact tracing. And if you want to make contact, then we are available. And finally, this is the main literature being referred to, but there is a lot of literature out there now, and they are getting better and better. You have to understand that even though it would be lovely to do these experiments, this is an infectious agent, and the experiments have to be done in such a way that you don't infect people. So thank you for listening to me, and thank you very much for this opportunity to share.
Thank you, Dr. Mitchell, for that class, Mask 101. We were taken to school on some very basic things, which might be common sense, but are rooted in science. After that, all of us, I'm sure you're sitting there thinking how much you have to reconsider how you are using and choosing your masks. And you know, as she spoke about that simple test that you use just to give an indication of if your mask is suitable. You hold it up to the light and assess how much light is able to come through. I saw one of our cameramen, Kadim. He took his mask out and he held it up and he began to assess the amount of light that was coming through. And what that said to me is that your talk, Dr. Mitchell, is already having impact on Jamaicans and everyone who is listening. Now, I'm not sure, Kadim, if your mask passed the test. I hope it did. But certainly, that talk is sending all of us to reevaluate how we choose and use our masks. So we are calling on all Jamaicans to be sensible and scientific about how you choose and use in this fight against COVID-19. Our next speaker, Dr. Debbie-Ann Gordon-Smith, is discussing a topic which is crucial to our safety. But like so many things with COVID-19, it is evolving in real time, and so little is known about it. She is discussing testing for COVID-19 in water and wastewater, significance and implications. Dr. Debbie-Ann Gordon-Smith is a graduate of the UWI and currently lectures in environmental and analytical chemistry in the Department of Chemistry, where she also manages the Environmental Research Laboratory. Her research focuses on the impact of human activities on Jamaica's marine and freshwater bodies, and involves the study of Jamaica's groundwater as well as discharges of freshwater to coastal environments. She also studies water resources management. She has a keen interest in investigating the effects of climate change on tropical water chemistry, and more recently, the analysis of water and wastewater to predict outbreaks and circulation of viral diseases within the population. Ladies and gentlemen, it is my pleasure to invite my colleague, another outstanding Jamaican woman in science, Dr. Debbie-Ann Gordon-Smith.
Thank you, Dr. Singh-Wilmot, and good afternoon, everyone. Today, I'd like to talk to you about an area of research in which I am particularly interested. For some time, wastewater has been used to monitor and track the prevalence of viruses and other diseases in wastewater through the population, and it is showing some utility in these current times. So in my talk on testing for COVID-19 in water and wastewater, significance and implications, I'm going to share with you some information on how the testing of water can be used in this fight against COVID. So some of the questions I want to answer today include, what is COVID-19? And of course, you've heard about this on many occasions by now, but I'll spend a very short time recapping. What is the role of chemists in this COVID-19 fight? How do we test for COVID-19 in water, since that's what we're talking about today? Is the virus present in different types of water? Is our water supply safe? And finally, how can we use water testing to help in this fight against the disease? So what is COVID-19? Well, as you're probably already aware, the COVID disease is caused by severe acute respiratory syndrome coronavirus 2, or SARS-CoV-2, or the novel coronavirus. The disease has posed a significant health challenge with massive human and economic consequences, with about 20 million confirmed cases and over 700,000 deaths worldwide. Jamaica is no exception, as we continue to deal with an increase in cases and deaths, with a concurrent hit on our economy. As you know, it is primarily spread from an infected person through nasal droplets and also via surfaces. And as we also know, the virus can survive on surfaces for up to 7 days. The disease typically shows as a moderate or mild to moderate illness, which may or may not require special treatment. However, in older persons and in persons with comorbidities, you can see a much more serious illness. So what does this virus look like? Again, I'm sure you've heard about this, so I'm not going to spend too much time on it. SARS-CoV-2 is a microscopic package of genetic material known as RNA, or ribonucleic acid. This is very similar to DNA, or deoxyribonucleic acid, and these are shown on the slide. If you notice, RNA is a single-strand molecule, while DNA is a double-strand molecule. And the molecules consist of a sugar molecule attached to phosphates, as well as some kind of nitrogen-containing base, and some examples are shown on the slide. So this RNA is packaged within an outer coating, which is a lipid membrane, a fatty or oily membrane. The virus itself cannot multiply outside a host cell, so what does it do? When it comes in contact with a host cell, it attaches itself to a particular set of proteins, then injects the RNA into the cell. Once inside the cell, the virus can then replicate using the chemicals, etc., inside the cell, thus making cells sort of virus-making factories. When the cells die, the virus is released, or multiple copies of the virus is released, and these can then go on to infect new cells. So of course I'm a chemist, so I want to spend a little bit of time on the role of chemists in this fight against COVID. In fact, chemists play a very important role in fighting many diseases, not just COVID-19. It starts from the analysis of the genetic material, or genome, which is then used to develop testing and detection protocols. We continue on to the development of surfactants and soaps, which we know can be used to wash our hands, to clean surfaces, etc., as well as disinfectants for sanitation, and let's not forget materials for the prevention of the spread. We're also involved in the development of drugs and vaccine and other areas of research and development, some of which you would have learned about today. Like others around the world, my colleagues and I in the Department of Chemistry at the UWI are ready and have already started working in areas to assist in this fight. There is a wealth of knowledge and experience in our department, and for many years, my fellow researchers have been investigating natural products and their benefits, drug development, occupational and environmental health research, research and development in industrial processes, material science, food research, and of course, environmental science. Which leads me then to the focus of my talk, where I will look at the detection of the virus in the environment, particularly in waters. So what I will do today is share with you some research that is going on currently around the world, looking at the testing and prevalence of COVID-19 in water and wastewater. I will try to provide answers to some questions that my own family, friends, and students have been asking me, and some of these are listed here on this slide. The first one I'm often asked is, how do we test for the SARS-CoV-2 virus in water? Has the virus been detected in various types of waters? Is water a route of transmission? Are our drinking and surface waters safe? What can we do to protect ourselves and the population? And finally, I would like to end with, can wastewater surveillance be used to help in this fight? So let's start with the first question. How do we test for COVID? Well, there are two main tests that we use for most viruses. One of them you've been hearing a lot about, I'm sure. This is the RT-qPCR test, and I will talk a little bit more about that soon. This is what we call a viral test, and this test detects the presence of whole or fragments of the virus, which means when the virus is detected using this test, the virus may be viable and able to infect others, or it may not be. Another test that can be used is the antibody test, and we tend to use this to tell if there has been a past infection. So for example, we would test for antibodies in the blood of patients. So what is this RT-qPCR test? Well, that is really short for real-time reverse transcriptase or transcription, Q polymerase chain reaction, and I apologize to the persons who did not like their chemistry in high school. Q refers to quantitative or real-time. But what does this involve? Well, as I mentioned to you before, the COVID virus is an RNA virus. To be able to detect the virus, we need to convert RNA to DNA in a process called reverse transcription. Once we have converted the RNA to DNA, then we can multiply or amplify the DNA by producing multiple copies and copies of this DNA using the process PCR or polymerase chain reaction. We use a number of chemicals in this part of the process in addition to the first part of the process, and then what we end up with is a signal that shows us whether there is a positive for the virus being present in the sample or a negative for the virus being present. So in the positive case, the virus is present and detectable. In the case of a negative test, the virus is either not present or below our detection limit. In RT-qPCR, the sample is treated with several chemical solutions to extract the RNA present in the sample, convert the RNA to DNA, and then, of course, to amplify the DNA and then detect it. Normally, the demand for the enzymes and chemicals that are used is relatively low and steady. So scaling up to the massive quantities that we now require for COVID testing takes a lot of time. In addition, the primers and probes that are needed for this virus are specific to the virus. So each set of primers and probes are produced for different viruses. It means also that we have to carefully prepare all of these reagents, all of the kits that you've been hearing about, and we have to ensure that these tests produce accurate results, meaning no false negatives and no false positives. As you can imagine, tackling all of these issues has been very difficult in these times and contributed to the delays we have seen in testing thus far. This is a test, as I mentioned before, that we use for most samples, including throat swabs, nasal swabs, saliva, and of course, water. So what are the findings? Well, to be honest, I have found more than 20 reported studies on COVID-19 in water and wastewater already. These are both peer-reviewed journal articles as well as preprints that have not yet been published. So I will try my best to summarize the findings here for the rest of my presentation. I have shown you on the slide, though, 4 websites that you can go to for some information. So good information can be found at these. And I particularly draw your attention to the last reference, which is a document published by the WHO on July 29, so quite up to date, which summarizes and lists most of the references that I use today. All right, so what are the findings so far? The first question is, of course, is COVID-19 virus present in feces and urine? Well, unfortunately, yes. So far, I have found more than 10 reported studies showing positive results for SARS-CoV-2 in feces and urine of infected patients. Most of these studies have found fragments of the virus which are not necessarily infectious, and it seems the virus is not so resistant to the GI tract as other viruses such as E. coli and other enteroviruses, and it appears to be broken down through the tract. A very interesting finding is the detection of the virus in the feces of patients up to 5 weeks after their respiratory samples tested negative, which suggests then that an individual who has been infected with COVID can continue to shed the virus for weeks after they no longer are sick or show a positive respiratory test. 3 of the studies found live infectious virus in the feces and urine of patients. What does this mean? It means then that fecal oral, as well as fecal respiratory and urine oral and urine respiratory transmission may be possible, which is not a good thing. What does this mean? It would be a very good idea to close the toilet lid when you flush. In addition, persons cleaning bathrooms of patients who have COVID need to be very careful. So, the SARS-CoV-2 virus has been detected in wastewater in at least 8 countries across the world, as you can imagine, based on what I showed you on the last slide. These countries include those that have shown a prevalence of the disease in the population, including China, the Netherlands, Italy, France, Spain, Australia, the US, as well as Brazil. Untreated sewage and primary sludge has shown the highest viral loads, as you can imagine, and the virus persisted up to 3 days in hospital wastewater and domestic sewage. Primary sludge, which is the solids that are separated off during primary treatment of wastewater, showed the highest viral loads. And that's likely because the viruses tend to adsorb or stick to solid materials, so you would expect to see the sludge containing higher viral loads. Somewhat lower viral loads were found in secondary treated sewage, and in the studies that were published, none detected the virus in tertiary treated sewage or disinfected wastewater, which is a good sign. Now, disinfection I'm talking about includes chlorination, or addition of peracetic acid. So what is happening? The virus appears to show low viability or stability in wastewater, and therefore, more than likely, low transmission. In fact, one of the studies showed a 99% reduction within 2 days of viral loads in primary sewage. Now, there are many factors that affect the survival of the virus, and this includes temperature, the presence of organic material and other solids, as well as the types of microorganisms that are present in the water. Very importantly is that the virus is detected in sewage weeks prior to the first reported cases. And there's an interesting study out of Brazil which demonstrated that. In this study, the scientists collected wastewater samples from back in November 2019 and saw positive viral loads. The virus did not appear in the population, or at least there were no reported cases in South America until 60 days later, and the first reported case in Brazil was 90 days later. So what does that mean? The virus had already been moving through the population in Brazil, but no one had presented sick or the testing had not been done. But the wastewater showed viral loads. In addition, increases in viral loads in the wastewater increased as the pandemic worsened. And as the viral prevalence in the population decreased, we also saw a decrease in viral loads in the wastewater. This is all very important. What does it mean? It means then that it is possible to detect the presence of the virus or the presence of infected persons in the population in wastewater before there is an outbreak or before even people know they're sick. So I'm sure you're wondering about this one. Are our natural water bodies safe? Well, once again, the virus has been found in untreated waters and rivers, and this came out of a study in Italy during the height of their outbreak. The source of the virus is still uncertain, and it may not or may have been related to discharge from wastewater treatment plants. Again, mainly fragments of the virus were found. But to be honest, other coronaviruses have been found in other rivers around the world, and they do not tend to survive well in the environment. Again, remember, the survival of the virus depends on a number of factors. What were the possible sources? As I mentioned, possibly from a wastewater treatment plant, untreated sewage, runoff, maybe sewage overflows during flood rains or other rainfall events, and even broken sewage lines. Again, since mostly fragments of the virus were found, there is probably a low potential for transmission. I leave that with a question sign though, because to be honest, we do not have enough data about this as yet, so we cannot draw those conclusions just yet. So a big question is our drinking water safe? Well, SARS-CoV-2 virus has not been detected in domestic treated water supply, meaning water such as we get in our pipes from the NWC. But what about other sources of drinking water, such as bottled water, trucked water? These are of more concern. And the reason for that is other sources of drinking water may or may not be safe, depending on how they're handled. So for example, domestic water is typically chlorinated. So by the time it gets to our house, the water is chlorinated and therefore disinfected and safe to drink. In the case of bottled or truck water, it depends on how they are treated. And even then, if they are disinfected, how is the water handled from source to the public? That is something that we have to think very carefully about. So what are my recommendations to water industries? We need to pay attention to good sanitation in these facilities, the handling of the water from source to public, including the truck drivers who are collecting and dispensing the water to the public. They need to wear gloves, masks, etc. to prevent any spread of the disease. What about pools and recreational waters, such as rivers and beaches? Well, the WHO has said that based on the current evidence, there is a relatively low risk of transmission, but again, remember, this may change. If a pool is properly maintained, meaning adequately disinfected, then there should be a very low risk of transmission, meaning chlorination of at least, or I should say free chlorine levels of at least 1 milligram per liter. But bear in mind that the risk increases in small crowded areas, such as in changing rooms, showers, and of course, the virus does live on surfaces, so we need to pay special attention and disinfect or sanitize the surfaces that may be touched by many people. So in conclusion, I want to tell you that SARS-CoV-2 virus, the COVID-19 disease, has been detected in waters of various types, including rivers and wastewaters, feces and urine. And in fact, the presence of the virus in these types of samples compare well to the prevalence in the population. The virus in untreated sewage may or may not be infectious, and there is no evidence of infectious virus in tertiary treated or disinfected sewage. Very important, disinfected sewage. Now, I cannot say that sewage treatment is 100% effective, but there is a significant decrease in viral loads after different stages of treatment. Sewage surveillance can be a useful tool that could be used by our public health authorities as an early warning tool to anticipate outbreaks, both on a local and regional scale, and also to map the circulation decrease or increase in infections throughout the population. And when I say a local scale, I mean as local as we could use it at the University of the West Indies by monitoring wastewater leaving dorms, for example. So even before persons may show symptoms, we could be testing the wastewater on a regular basis to catch any early infections in these dorm rooms. There is one limitation, of course. This sort of testing or use is really only applicable in sewered communities, meaning communities that are connected to the central sewage line, or where you can go and collect the wastewater as the water is leaving those communities. When it comes to rural areas and other places that are not sewered, we'll have to get a little bit more inventive with that sort of testing. So what can we do? Well, it's not all doom and gloom, but I want to tell you first of all that we need to protect our drinking, recreational water sources, which means we need to avoid disposing of fecal matter or sludge or untreated wastewater into our water bodies and around our water bodies, meaning our rivers or seas, etc., especially where persons are using the water for domestic use or if they're using it for recreational use. My advice to you is to drink treated, disinfected water, and remember that trucked, bottled water, or stored water for a period will also need special attention. So if you're storing water for an extended period, consider additional disinfection, such as adding chlorine bleach, or if you're going to drink the water, boiling is also very effective at killing the virus. My recommendation for water industries is to employ proper sanitation practices, PPE for your staff, and start doing regular testing. We know that viral loads in sewage decrease at each stage of the treatment, but we should consider adding a disinfection step before discharging the water into our water bodies. Infected persons should use separate toilet facilities where possible, and of course, fix all faulty plumbing and sewage lines that may be broken. Finally, sanitation services and workers must be careful when they're handling untreated or treated sewage. Again, personal protective equipment, safety precautions need to be taken, and personnel need to be trained and briefed on how these procedures need to be employed. In addition, we know already that wastewater technicians are often tested for other diseases. We may now have to include COVID-19 testing, at least I would recommend it. And this is a part that UE can play a role in. We should start surveillance of SARS-CoV-2 in wastewater, in sludge, and in our water bodies, such as our rivers or groundwater, especially those that are impacted by untreated sewage. At the UWI, this is one way in which we aim to fulfill the needs of this society. To do so, we are seeking partnerships with non-government and government sectors, as well as international partners to meet this goal. My colleagues and I in the chemistry department at the UWI are ready to assist in this area and other areas of COVID research and action. So, that's it for me today. I hope you found my talk on chemistry interesting, and I thank you for listening, and stay safe.
Thank you so much, Dr. Gordon-Smith. So much there to think about. So much environmental monitoring, so much research data that is needed, so much for us as scientists to do to protect our country and our region. You have brought into perspective the overall need for proper sanitization practices, not only by healthcare professionals and in our healthcare facilities, but also in our communities and in our homes. We certainly thank you for calling on those who manage our water supplies to engage proper standards, and we also thank you for bringing this awareness to us. I'm sure many of us before now had not thought about COVID-19 in water and wastewater. We hope that you and your research team in FSD can attract the funding, both locally and internationally, to do this kind of monitoring and to keep us safe, to use the science to fight COVID-19. Our next speaker was chosen, ladies and gentlemen, because his work in his country shows that collaboration accelerates the progress of science and deepens its impact. This is especially true for the least developed countries like Jamaica. And in this fight against COVID-19, the researchers here in FSD embrace cooperation and endorse a multidisciplinary, multi-sector, multinational collaborative approach. Dr. Rolando Gittens Ibakashe is our speaker in this section on international scientific collaboration, an immediate imperative. His talk is entitled Preclinical Validation of a Locally Produced Surge Mechanical Ventilator for COVID-19 in a Developing Country with Limited Resources. Dr. Gittens is a research engineer at the Institute for Scientific Research and High Technology Services in the CASAT AIP in Panama. He holds a bachelor's in electrical and electronics engineering from the Technological University of Panama, a master's in material science and engineering, and a PhD in bioengineering from Georgia Institute of Technology. Dr. Gittens is a member of TIAN, the Young Affiliate Network, which is an agency of the World Academy of Sciences, TWAS. And it is through our membership in this network, I am also a TIAN member, that some of us here in the FSD have come to know and work with Dr. Gittens. His research focuses on the role of nanostructural and electrical properties of biomaterials for tissue regeneration, and has resulted in one patent and another active application for the surface modification of titanium implants. Dr. Gittens actively applies his soft skills in intellectual property and science diplomacy for scientific lobbying to enact laws that support the science, technology, and innovation systems. And he is also a consultant for business innovation through research and development. I call on all the Jamaicans and the non-Jamaicans in the audience to give a warm Jamaican welcome to Dr. Rolando Gittens.
Hi, my name is Rolando Gittens. I'm a research engineer in Indica Sat IP here in Panama. And I wanted to thank the organizers, Dr. Marvadeen, for the invitation to participate in this amazing forum series that the University of West Indies is organizing. And it's just a pleasure to share with all your community and with the region about what we're doing down here in Panama. So I'm going to get started, and the title of my talk is International Scientific Cooperation, an Immediate Imperative. But as I discussed with Dr. Marvadeen, I will try to give you an overview of the research that I'm doing here in Panama, focused on regenerative engineering and biomaterials, how international collaboration has been key for me as one of the few scientists working in this field here in Panama, has been able to survive, but then also how once the pandemic started, everyone had to reinvent themselves, and we all had to find ways to support the health authorities and the government in providing all the reagents and materials needed for the country response. So with that, of course, I need to start by giving you some very quick words about what Indica Sat IP is here in Panama. And Panama is probably best known for many other things other than science, at least, for its coffee, for Roberto Duran, for its booming city that is growing every day, for its amazing biodiversity, and of course, for the Panama Canal, especially now that it has its expanded locks. Well, Indica Sat would like Panama to also be known for its scientific productivity. And we happen to be actually right in front of one of the Miraflores locks here in front of the canal, in a relatively new building that has been designed to do research and have all the necessary spaces to do so. So Indica Sat, and without going into too much detail, is basically a national research laboratory that is focused on three main areas: biodiversity, biotechnology, and biomedicine. And for that purpose, we have around 25 scientists in different fields, from chemistry and pharmacology, neuroscience, and, I guess bioengineering with me, trying to work on different areas and collaborating, trying to solve different problems. And so the particular research that I'm focused on, or that I'm interested in, addresses an important issue related to the elderly population and the aging of the population worldwide. In Panama, at least, I'm showing you here some of the population pyramids from 1950. You can see it's basically a perfect pyramid, like you would see on a textbook. But by 2020 and what's expected to happen by 2050, you can clearly see that there's a huge distortion on the pyramid, basically caused by the increase in the elderly population. And just for fun, I also look for some of the information in Jamaica, and you can see that the story is pretty much the same. And this is happening around the world. And the situation with this is that, the good news is that because of technology and because scientific advances, we've been able to find new treatments and new ways to allow people to live longer lives. But of course, we don't just want to live longer, we want to live a better life. And so there are many diseases and problems that can become an obstacle to that objective. And so some of those diseases that we study in my group, or that we focus on, are osteoporosis, stroke, and cancer. And so the perspective that we take to try to tackle these problems in our group is focused on regeneration. And regeneration has been a topic that has been discussed for a long time, from the Greek mythology and Prometheus, who was punished by Zeus by sending an eagle to eat his liver every day for eternity. And the interesting thing is that the liver is actually one of those internal organs that does show some capability of regeneration. But going away from mythology and going back into biology, there are several organisms and animals that show remarkable capacity to regenerate. And I don't know how many of you know this little animal, but it's a species of salamander called the axolotl, and it's endemic to Mexico. And one of its properties is that if you cut one of its limbs, in a matter of days, it forms what's called a blastema, which is this green blob here. It's a mass of cells that, where there used to be skin and muscle and bone, these cells start going through what's called a de-differentiation process to turn into stem cells. And then in a matter of weeks, then these cells are able to regenerate and reconstruct all the different tissues that were there before. And so the idea is that several animals, including humans, we have some innate regeneration capacity. And the question is, how can we unlock those doors, and how can we allow our bodies to regenerate by themselves? And so that's what we're studying in my group through the development of new biomaterials that can help this process of regeneration. And so we've done a lot of work with titanium implants for bone applications, and more recently with some soft materials. But of course, it's not just a matter of building or producing these new biomaterials, but we also develop in vitro models that allow us to test if these biomaterials are actually able to do what we hope or what we expect them to do. And then we also use different bioinformatics and imaging technologies to try to characterize these regeneration processes. So I'm going to give you two quick examples of how we've used regeneration or this idea of biomaterials for regeneration. One focused on bone and the other one focused on brain. And the work that is related to bone starts from my time during my PhD thesis in Georgia Tech in the United States. And the focus there was how can we improve implants that have to be applied to bone? Now, interestingly enough, bone is a very complex material itself. It has a high degree of complexity at the microscale level, and that's why we have so many different bones in our bodies with different shapes, each one doing a very specific job. But we also have a lot of complexity at the microscale level due to the different type of cells that are present in bone. Like the one that you see in the image is called an osteoclast, here marked with an OC. And the job of the osteoclast is actually to go to areas of bone that have gone through injury or wear and tear, where there is presence of microcracks. And so these cells go there and have to resorb the bone or degrade the bone in that area. And interestingly enough, it's exactly in that area where the osteoblast, which is a different kind of cell that produces the new bone, goes and produces the new bone right there. And so the question is how these osteoblasts detect or know exactly where they have to produce a new bone. And so one of the suggestions is that the combination or the hierarchical complexity of the micro and nanostructure surfaces, because these osteoclasts are not able to remove the protein collagen tufts that are on the surface. So you can see this very intricate nanostructure on the surface of that bone surface. And the idea is that maybe osteoblasts are capable of recognizing some of those structures and produce the new bone in that area, besides, of course, the biochemical information that is there to tell them what to do. So that's basically have been the career or the job of my advisor in Georgia Tech, Dr. Barbara Boyan and Dr. V. Schwartz. They've been studying these for decades, and they've been able to show pretty conclusively that the surface properties of titanium can impact and can promote bone formation. And so in my particular case, my focus was what's the role of nanostructures on this process, or nanotechnology. And so schematically, you can think of it as the bone interacting with the implant at the microscale level, and that's why most implants have the shape of a screw or have some type of design that gives it a good primary fixation. But then you also have to think of the implant at the microscale level, or at the level of the cells, where they have to have enough roughness so that the cells feel like they know where they are, and they have spaces where to grab on. But then we also have to think about it at the nanoscale level. And at the nanoscale level, you can think of the proteins that absorb on the surface, right? These are proteins that are either in saliva for dental implants, or in blood, or any other biological fluid. They're going to start interacting with the titanium surface, and directly and indirectly, they're going to interact with the proteins on the proteins on the cytoplasmic membrane of the cell. So that's why it's very important that we think of surfaces and biomaterials at these hierarchical levels. So having this in mind, we've developed novel nanomodifications for titanium surfaces. And you can think of a titanium implant that comes out of the machining after being produced with a surface that doesn't have that much geometrical features that you can very easily describe. So for our purposes, this is a relatively smooth surface that cells really don't have much to interact with. But after applying our nanomodification techniques, we can turn the surface into something like this, which gives it a lot more surface area, but also a lot more complexity to interact with these proteins, like I showed on the schematic. So we've been able to show that these nanomodifications can be applied to complex 3D implants, like in the case of this spine implant made out of titanium. And you can see that our modification is a no line of sight modification, meaning that you can modify both the outer surfaces of the implant, but also internal surfaces that may be very difficult to reach to with other different type of modification techniques, like grid blasting or something like that. And of course, after characterizing the material, we've also done lots of studies trying to evaluate how these cells interact with the nanomodified titanium surfaces. And what you see here in purple, it's an osteoblast interacting with a titanium surface that has been micro and nanomodified. And what you can see here is that the cells are able to interact both with the microstructure, but also with the nanostructure. And of course, we've been able to show this not just qualitatively with SEM micrographs, but also using different techniques like ELISA and PCR. So the good results and this concept that we basically were able to prove on bone surfaces, showing that if you control the surface properties like roughness and surface energy and the nanostructures on the surface of titanium implants, you can improve the differentiation of these osteoblasts and produce bone that is better quality bone. We wanted to take that same concept and apply it to different tissues. And so, of course, for the project related to titanium implants, we continue to collaborate with my mentors now in Virginia Commonwealth University and other researchers in the United States. And so international collaboration to be able to do this work from Panama has been key. A lot of the equipment that you need to characterize these surfaces and to do these studies are very expensive equipment, and it's especially hard to justify purchasing some of this equipment just for a few scientists that we have down here in Panama. So international collaboration opens a lot of doors to be able to push our research forward, having access to some of this equipment in other universities, and also, of course, applying for different funding and getting new ideas running. So like I was saying, we've taken the same concept and applied it to brain regeneration. And the main application here, in part due to a great collaboration that we started with Dr. Miguel Perez Pinzon, who is a Panamanian that has been working and living in the United States for over 40 years, and who is the director of the Center the Cerebrovascular Disease Research Center in the University of Miami. And basically, he is a specialist in stroke. And stroke is a very complicated disease in that it affects a lot of people. It's one of the major killers around the world and here in Panama. And the major issue with stroke is that when you have a blockage of one of the blood vessels in the brain, you then have a region of the brain that ends up lacking access to oxygen and nutrients. And the neurons in the brain and all the types of cells that are in the brain are so energy intensive that even just within a few minutes to a couple of hours, that these cells don't have access to these oxygen or nutrients, they start dying. And so right now, the current options that patients with stroke have is basically going to the emergency room as quickly as possible so that doctors can remove the ischemic attack, or try to provide some other type of treatments to try to take care of the hemorrhagic attack in the case that it's a hemorrhagic attack. But the reality is that basically 20 to 30% of the patients are able to get to the hospital within that acute window of treatment, which is 2 to 4 hours. More than 70% of the patients actually end up arriving to the hospital later than that. And when that happens, they end up going into the chronic stage of the disease where there starts to happen a chronic death of neurons in that area. And right now, there are very few options that are able to address this chronic neuronal death. And that's one of the main issues with recovering from a stroke, because the difficulties that patients have to regain some of the motor and cognitive deficits. So the idea here is that we want to try to apply a similar approach where we develop a biomaterial that can interact with stem cells and that can hopefully try to stop some of that neuronal death in that region where the stroke happened, and in the best case scenario, try to revert some of the damage already done. And the idea would be also to develop this biomaterial so that it can interact at the microscale level with the cells, but also at the nanoscale level. And so, to give you some background on this part of the project, brain is a very complex organ, as you know, or as you can imagine. There have been some amazing studies coming out from Brazil trying to evaluate the composition of cells in the different subregions of the brain. And in this study, they divided the brain in 3 areas: the cortex, the cerebellum, and what they call the rest of the brain. And you can see that the brain as a whole has about 86 billion neurons and 84 billion non-neurons. And that, in general sense, gives you an idea that there's about the same number of neurons as other type of cells like astrocytes and microglia and other types of neural cells. But if we start breaking it down by region, when you look into the cortex, you can see that the cortex has about 16 billion neurons, but 60 billion non-neurons. So there's a lot more non-neurons, so other type of cells, than neurons in the cortex, versus, for example, the cerebellum, where we have around 70 billion neurons and only 16 billion non-neurons. And the cerebellum is one of the smallest parts of the brain, so it just accounts for 10% of the brain mass. So you can imagine that these separation or these differences in cell populations in the different brain subregions also demands a different composition in the extracellular matrix that allows these cells to do the job that they have to do. And so we've been studying a little bit about that because focusing on the biomaterials and the interactions that cell has with its environment, if we're going to try to develop a treatment for stroke in a particular area of the brain, we need to know exactly what kind of properties our biomaterial should have. So what we are using for this project in particular is a technique called decellularized extracellular matrix. And what it involves is that, again, you have a brain with all its different subregions, and we process the brain and separate these 3 different regions of the brain and then pass it through a process of decellularization, where the main objective is to remove the cellular component, but leave behind that extracellular matrix where the cells were attached, and use that extracellular matrix, now that it's been decellularized, as a biomaterial. And this is not a new idea. I mean, there's a lot of even commercially available products that are based on decellularized extracellular matrix. Some are made out of intestinal submucosa from pigs. Some others are made out of placenta membrane from humans. But in this particular case, we were interested in the brain. And there are several studies that show that the closer your extracellular matrix resembles the tissue that you're interested in regenerating, the better results you can get. And so in the case of the pig, there wasn't that much information available. And so a big portion of this project was performed by Dr. Diego Reginensi, who was a postdoc for this project. This project actually was financed through a grant that we received from NIH, again, thanks to that collaboration with Dr. Miguel Perez-Pinzon from University of Miami. And one of the things that we were able to show is that in the pig brain, we see a very similar behavior, where most of the neuronal cells, or the cell population in the brain, was found in the cerebellum. As you can see here, the percentage of cells, or the number of cells, 55% of the cells in the brain were found in the cerebellum, where only 25% were found in the cortex, and about 20% in the remaining areas. So this was very important information for us, because what we're trying to do is exactly that, remove the cellular component from these tissues. And so knowing what we were starting with was key for this process. So very briefly, and just to give you a quick idea, what we do is basically process the brain, separate it into the three different regions, decellularize each one, then we lyophilize the tissue for better handling and processing of it. And of course, we always did some microbiological assays to make sure that we were starting with a sterile aseptic tissue. In very broad and general terms, the main objective of our studies were to test how, in this particular case, a cell line called PC12 cells would react to the different biomaterials from the different brain regions. And you can think of the control group being like panel A, where cells grow in very spherical colonies without much activity or differentiation. And when they are exposed to our biomaterials, we would expect some type of differentiation, like the one that panel B is showing, where these cells are able to extend these axon-like structures that exhibit or reflect that these cells are in a conducive environment for neuronal maturation. And so in a snapshot, the main results of this study that we published earlier this year is basically that, we had our control group, so the group that was not exposed to anything. We had some positive control groups that were exposed to different neurotrophic factors like nerve growth factor or BDNF, and we show clearly how these cells undergo this maturation process. And then we evaluated how our different biomaterials, using different decellularization protocols, one that lasted for 4 days and one that lasted for 1 day. And we show here in the quantification that indeed our biomaterial is able to differentiate these PC12 cells, maybe not to the extent of the positive control groups, and we believe that that may be an issue of concentrations, but they were all significantly higher than the control group that did not have any stimulant. And so this was very exciting results, because we are showing that our biomaterials are able to create this environment that is conducive for neuronal differentiation. And again, in collaboration with Dr. Miguel Perez-Pinzon and his team, now we've been able to use these biomaterials with primary neuronal cultures, which are very difficult to maintain, and show that our biomaterials also are capable of culturing these primary neurons. And interestingly enough, show that there are some very interesting organizations of the cells on the surface that we're still evaluating if they're just artifacts or if this is actually some physiological response that we're finding. So that's most of the research that we've been doing for these last few years and what we've been busy with here in Panama. But starting in March, when we started, hearing about the first case of coronavirus in the country, Panama has actually been one of the hardest hit countries in the region. So I imagine in Jamaica, and I was looking at the numbers, and you seem to have been doing actually amazingly well with this whole situation. But here in Panama, this is basically one of the very common views. We still are in a very strict quarantine. So the few spaces where there are people allowed to be outside, everybody has to be wearing their face masks. Another very common scenario, something that probably many of us has had to go through, is the nasal swabs, so that we can get tested and evaluate if we're positive or not. So all part of this new reality. And sadly, of course, there are many, many people here in Panama. There's over 1,500 people that have died because the coronavirus or the COVID-19 disease hits them very hard. And so these patients with very severe conditions end up having to go to the intensive care unit and have access to all these different equipment and treatments to be able to survive. The sad part is that for a small country like Panama, that it's used to import a lot of its technologies, we actually are in a very dire position, because right now there's a global shortage of many of these equipment and many of these materials, even for the sampling, even for the testing and the collection of the samples. So that's where a request from the Ministry of Health and our, Institute Gorgas, who is in charge of public health research here in Panama, requested the help of IndicaSat to produce these viral transport media that is very needed to put the swabs after the sample has been taken, and so that it preserves the sample intact until it reaches the laboratory, and it enhances the chances that the sample gives an accurate result. What started from a very rudimentary process, trying to get this media prepared in IndicaSat, has gone through several iterations, and now we have a little bit of a better presentation. But I'm very happy to say that, starting from the beginning of April, IndicaSat has been able to provide some of this media to the country and share these so that the country does not let the diagnostic program go to the ground and continue to do the testing that is so necessary to respond to this virus. And so just in a very quick series of graphs, we can show you that at least for the first 2 weeks of April, Panama had to depend strictly on the media that was produced in IndicaSat. And we can show that after validating it with the Institute Gorgas to make sure that it worked as it was expected to work, we see here that in the national response, the media that was produced was able to do the job and allow the country to have the diagnostic that was needed. So, of course, the media is one component of the sample collection, but the other important aspect of sample collection is access to the swabs, and the swabs are also in global shortage. So we've been working in collaboration with Dr. Guido Esquivel, who is a maxillofacial surgeon here in Panama and is an expert on 3D printing. And so we've been able to 3D print our own nasal swabs that right now we're going through some testing to make sure that they behave just like the commercial ones that you can find in the laboratories. And of course, we're also preparing a protocol to make sure that we evaluate how it behaves with a series of patients before this is able to use widespread in the country. But we think that this capability of producing our own materials is mandatory, and we think that even in the mid to long range, it will be very important so that the country can have access to the materials that they need. And lastly, and not to take too much of your time, but I wanted to share with you another experience that we're having producing ventilators here in the country. That's another one of those equipments that is in global shortage. Countries are prohibiting the exports of this equipment because they also need them, and they're also suffering the impact of the pandemic. And so we started also from the beginning of April, helping these different teams of engineers that started working on the development of prototypes of ventilators, something that had never been done in the country before. So we don't really have research established or real expertise established in the development of mechanical ventilators. But it's just been an amazing experience to see how so many institutions, so many different people, luckily so many doctors in the little free time that they have from the intensive care units, to offer their knowledge and to help develop these devices in a matter of a month, in a matter of 3 months, already having at least one prototype that has gone through the whole process of validation, including preclinical testing with animals. And so we have been able to show that these equipments are able to produce and behave as expected and maintain these animals well ventilated for over an hour, including animals that have gone through a process of, through a model of lung injury to try to simulate the breathing difficulties that a patient may have with COVID-19. So here are some of those results. Basically, with the healthy animals, we show that at least in the parameters that are related to the oxygenation of the animal, the control, which is a commercial, ventilator that you would find in one hospital, the parameters are basically exactly the same as with the prototype. And again, this is after ventilating for at least an hour with the prototype. And after the lung injury, here we show how we established the lung injury. You can see how the peak inspiratory pressure increased considerably, how the levels of oxygen in blood went down to very critical levels. And then after connecting the animals to the prototype, we see that we're able to rescue these animals and increase the oxygen levels to reasonable levels of oxygenation and comparable to the levels of oxygenation that the control ventilator provided. So with that, I just wanted to thank one more time Dr. Marvadeen and the University of the West Indies for this opportunity. Of course, all the institutions that have provided funding and support for these different projects, especially now with the projects related to COVID-19, there have been so many different companies that even if they have not been able to provide funds directly in money, we've been getting loans of equipment, donations of the animals, so that we could do these experiments and show for the first time in the country, and also with international collaboration, I must say, because especially for the regulatory component, we're working very closely with Mexico, the COFEPRIS, with Virginia Commonwealth University in the United States, and with a big network of metrology laboratories to try to make sure that we comply with everything that is needed, and so that these equipment are as safe as possible for patients. And with that, here's my email, and I'll do my best to try to answer some of the questions that you may leave on the chat. So thank you very much for your attention.
I thank you so much, Dr. Gittens. We commend you and your team for this innovation. Ventilators for Panama does remind us that cooperation among scientists is necessary to put science in action and fight COVID-19, especially in resource-limited countries like Panama and Jamaica. Now, FST students have been cultured in a space right here in the faculty, which promotes science as a tool for innovation and problem solving. So we move from Dr. Gittens' ventilator innovation to ventilator solutions right here at home, solutions that our students are creating to fight COVID-19. It is my very great pleasure to introduce to you a young man whom I have watched grow right here in the Faculty of Science and Technology, Mr. Yekini Wallen-Bryan. Mr. Wallen-Bryan graduated from the FST with a double major BSc in Electronics and Energy and Environmental Physics. He is currently still with us, pursuing an MPhil in Electronics Engineering. In the final year of his BSc program, Mr. Bryan got heavily involved in several internships and projects, On a part-time basis, he was conducting energy audits for a local firm. He gained technical experience with a large local telecommunications company, and he led and worked on numerous technical projects for the university and other entities. Since graduating, Mr. Wallen-Bryan went on to form Prelabs Limited in 2016. He and his team at Prelabs have implemented several projects and have developed several award-winning products such as PowerPre, a line of devices and an app that allows people to monitor and control electrical appliances from anywhere in the world using their smartphones and computers. I'm sure he will tell you some more about that. He has attracted the support of several organizations and private companies, such as the Development Bank of Jamaica, the World Bank, the Caribbean Climate Innovation Center, and Total Jamaica. Please welcome this young scientist, innovator, and entrepreneur, Mr. Yekini Wallen-Bryan.
Hello everyone, my name is Yekini Wallen-Bryan, CEO and founder of a company called Prelabs Limited. What we do at Prelabs Limited, essentially we're a product development company that's centered around remote monitoring, control, and automation. So we develop products and we make custom solutions around these things. We specialize in Internet of Things type solutions. So we develop PowerPre, which is our flagship product. It's a system comprised of devices, smartphone applications, and a web page that allows people to monitor and control their electrical appliances and energy consumption overall from anywhere in the world, from their smartphone or their computer. Just to give a bit of a context about what we do mainly. So on screen are a few videos of a couple of our applications. One of them are an example of one of our solutions turning off a gate light from a smartphone. Another one is an Airbnb, where as you enter the room, the air conditioning and the lights turn on automatically, and you can turn them off via smartphone as well. That's just a bit of a context as to what the PowerPre solution itself does. Essentially what PowerPre does in terms of the value that it adds, it allows people to see their energy consumption in watts as well as dollars. It allows people to control their appliances from anywhere. It allows people to automate their processes so that they don't have to remember to turn things off anymore. It just does it automatically based off of motion sensors or time schedules, which allows them to save on their energy bills. It provides them with analytics to help them understand exactly how well they're performing in meeting their targets from an energy consumption perspective. And it's a complete package. We have a device for just about every type of appliance in the home or office. Since launching PowerPre, we've expanded to be able to have a smart complex that's on Dewsbury Avenue, where every unit that is going up has our technology built in. Owners from anywhere in the world can see the consumption of the Airbnbs in real time and know that their air conditioning units and lights will be turned off automatically if no one is inside the room. We went into this venture because we recognize that smart buildings are the future, and the future is now. As we approach a sustainable future, both climate and otherwise, it's important for us to be able to ensure that we can use our resources more effectively. We have a certain version of this product for businesses as well that allows them to break down the data that our devices collect on energy consumption in a way that helps them perform more practical energy audits and financial audits as a result. So we've taken the same technology that we've applied to energy and are now applying it to water. With WaterPre, as the name suggests, it's allowing you to pre your water, as the Jamaican colloquial term, and PowerPre is essentially allowing you to pre your power and your energy usage. WaterPre is comprised of three main components: flow sensor, water tank sensor, and a motorized or electronic valve. WaterPre allows people to be able to have up to 40 milliliters of accuracy and understanding of what the consumption in terms of water is, whether it be for their home or their business place. And if they have a leak, which of course is well over 40 milliliters, they have the ability to turn off the water coming in to the supply of their home or office from their smartphone. Whether it be the case of an emergency or a faulty appliance, people are able to have a better grasp in understanding this black box about water consumption and not be surprised at the end of every month when they have an exorbitant water bill or excessive water damage that they weren't aware of. The last part, again, is a water tank sensor, which allows people to see exactly what's happening inside the tank instead of having to go on a roof and knock on it to figure out what the level is. So this is just a nutshell about the type of solutions that we've been building. Yeah, we've developed custom solutions for other clients as well, scientific and industrial SCADA systems that allow people to, again, to be able to monitor and control different parameters about processes to help them understand exactly what's going on. So that's just a bit of a precursor about what we do at Pre-Labs. We've been getting a lot of recognition recently for the type of stuff that we do. Of more relevance, since the start of the COVID-19 crisis, we've been trying to figure out different ways in which we could apply our resources and time to figure out what we can do to help mitigate some of the effects of this pandemic on our healthcare system and our broader population. At the start of the crisis, I got a call from a friend asking if I knew how to build a ventilator after learning that there was a shortage locally. I didn't know anything about it at the time, but I told him I'd look into it. Saw that there was a huge global effort going into actually making low-cost emergency ventilators that could apply some form of ventilation to people in hospitals, patients that have COVID and otherwise. So we thankfully were able to get going on that. A lot of things blossomed outside of that as well. We saw that we had the opportunity, given my experience and access to 3D printing, we started printing face shields, 3D printed face shields, of which we've donated over 2,000 to the healthcare sector. Thanks to the support of a lot of our corporate donors, we've been able to procure medical grade 3D printers as well that allow for the printing of special types of valves, adapters, ventilator splitters, and even COVID-19 testing swabs, FDA approved COVID-19 testing swabs. Outside of the Citizens Response Group, which is a non-profit entity, we've also been producing hand washing stations commercially at the request of the Prime Minister to local BPOs and health centers, automated contactless hand washing stations. You put your hand there and it dispenses water and soap automatically without you having to come in contact with anything, as you can see on the screen. Also on the screen are about 300 of the FDA-approved testing swabs that we've been able to print as well. So we're a young company. We're a new company, young not just in age, but our team also is comprised of a lot of individuals under the age of 30. However, our output in terms of what we've been able to produce speaks volumes. We've been able to provide solutions that otherwise is not accessible or available in the context of the Jamaican region. Our approach to being able to do any of these things are what we like to say, plan like an engineer, reason like a scientist, and create like an artist. When I say plan like an engineer, we really mean try and think about everything that could possibly go wrong and come up with a plan to deal with it. Always have a plan A, B, and C, and ensure that things are always in your control in some way, shape, or form. When we say reason like a scientist, what we mean is apply the scientific method to every aspect of not just development, but also business. You know, whether it is a case where you're trying to get market research or you're trying to conduct a market study on a product to determine the true value proposition and exactly how many people would want it, to understanding your projections and being able to make concrete assumptions about exactly what to expect. It's important to understand the scientific method to be testing your hypothesis, whether it be through primary or secondary research, and coming up with a conclusion that is derived off of facts and data. And when I say innovate like an artist, I really mean that when we try to approach a solution that, for example, when a customer comes to us, a potential customer or client, and expresses a problem that they're having, that they're not able to find a solution for, we have to be able to step outside of the box, think about all the resources at our disposal, and figure out how is it that we can apply these resources to solve their particular problem. To do that requires a certain level of creativity that we like to liken to a form of artistry. Definitely when the solution comes to the fore and you see something that is adding value to someone that is reliable and works consistently and works in the eyes of the customer beautifully, that in itself is a form of art in our opinion. We do all of this by knowing our foundation and applying where necessary. Once we have a solid understanding about the nature of how things work from an electronics, from a software, perspective, then we're able to apply it to whatever the relevant context is. And above all, we have to ensure that we're practical with regards to our time, of which we have a finite amount, the objectives that we set, and the commitments that we make. Nothing that we're doing makes any sense unless we're able to understand exactly what it means and what it takes to deliver on a solution and to ensure that it actually gets delivered in a timely and consistent way. So myself, I am a graduate of FST. I did electronics, a double major in electronics and energy and environmental physics. And all of our technical team right now are also FST graduates. We're also all former robotics team members. Being in the space, in the faculty, in the physics department, provided a context for us to really apply what we know, to step outside the box and think of creative ways of solving all different types of solutions. And this has allowed us to apply that same thinking to try and solving some of the problems that society is actually facing in a way that hasn't been done before. We have a lot of mentors in the FST space that have always believed in us and pushed us to try to do things outside the box, things unorthodox, and to solve problems. For this, we're very grateful. I would also just want to close by mentioning that science has to be the way forward in all sectors of society if we want to approach any meaningful goal of sustainability, improvement, and development. So thank you, FST. Thank you, audience. That's it for my presentation.
Thank you, Mr. Wallen-Bryan. You know, I felt it in Forum 1. I told you so, after the student response. And today, I felt it again, a regenerated spirit of hope as I saw the future of science and the future of Jamaica in good hands. I heard your tagline, Mr. Bryan, plan like an engineer, reason like a scientist, and innovate or create like an artist. And I want to take you on that. What you just described really is, in fact, the scientific method. So I want to say, plan like a scientist, reason like a scientist, and create like a scientist. However you want to say it, though, ladies and gentlemen, STEM, call it STEAM, call it what you may. It is science, science creating, science solving problems, science translating into wealth, science translating into business, science making people's lives better, science in action to fight COVID-19. A commendable enterprise, a commendable response from our students here at FST. And that, ladies and gentlemen, is a wrap for Forum 2 of Fighting COVID-19 Science in Action. Now, if you found any of these talks informative or useful, please pass on the word by sharing this video with others who could not make it today. I want to thank all the speakers for their work and for taking the time to share their work with us today. Dr. Ricardo Anderson, who showed us the interdisciplinary nature of data science and how it is crucial in the fight against COVID-19. Professor Rupika Delgoda, who showed us the potential of Jamaica's natural products, Jamaica's gifts from nature. She shed light on their potential in the fight against COVID-19, and she guided us on how to safely use these gifts. Dr. Sylvia Mitchell, ladies and gentlemen, who has merged science with common sense to give us important guidelines to choosing and using masks. Dr. Debbie Ann Gordon-Smith, who shed light on the need to monitor water, both wastewater and drinking water, in the fight against COVID-19. Our colleague, Dr. Rolando from Panama, who illustrated that scientific collaboration can help us to overcome challenges and position developing countries like Jamaica to fight COVID-19. And today's rising star, Mr. Yekini Wallen-Bryan, who showed you how our students here at FST, how our young Jamaican scientists are innovating, putting science in action to fight COVID-19. Now I want to say thanks to the people who worked hard behind the scenes to bring this to you. Thank you to the hard-working production team at UETV, led by Mr. Jamani Dunn, his team members, Mr. Kadeem Lawrence and Mr. O'Dane Thompson. Their professionalism and talent are responsible, ladies and gentlemen, for bringing this event to you today. Thanks to the External Engagement Committee in the Faculty of Science and Technology for organizing this event. Special thanks to the chairman of this committee, Dr. Andre Coy, for his leadership and the hard work of Mrs. Terry-Ann Collins-Frey and Mr. Maxwell Williams. Thanks to the dean of our faculty, Professor Michael Taylor, for the vision he had for this forum, and to all other colleagues in the faculty for their support. Thanks also to the moderator of our online chat, Dr. Kimberly Stevenson, for coordinating this interaction, which is so crucial to this process. And thanks, ladies and gentlemen, to all of you who are tuning in from all parts of the world. Thank you for coming. Thank you for participating. Please look out for the final installment of Fighting COVID-19 Science in Action. And please put all that you have learned here today in action to fight COVID-19 as you go about your daily activities. Thank you all, and please stay safe.